Hi everyone, I'm Brock. So today let's review an academic paper I have read this for this week. This paper is called AlphaFold 3: Accurate Structure Prediction of Biomolecular Interactions. And this is the original paper. So actually, this paper is not very long, huh? Oh, no, it's pretty long. Okay. And let's see. So AlphaFold 3, AF3, is a deep learning model capable of predicting the 3D structures of nearly all biomolecular complexes. That's insane. It can predict not only protein structures, but also interactions between proteins, nucleic acids, DNA, RNA, small molecular ligands, ends, and modified residues. That's crazy. If we can do this efficiently, it's really possible that we can cure a lot of disease, which means that some disease we cannot deal with today, maybe, just like get a cold 10 years later. Let's see how it works. Feature 1, protein-ligand. Well, I guess that's how to pronounce it. I need to say.
Ligand.
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 Ligand. Protein-ligand interaction prediction. Imagine designing a key drug molecular
Molecular.
molecular
Molecular.
molecular
Molecular.
molecular drug molecular to open a lock protein. AlphaFold 3 can predict what shape the key should be to fit perfectly into the lock. Interesting. So the input will be amino acid sequence of protein, PORCN, and the chemical formula of drug, LGK974. The output is predicted 3D structure of how this drug binds to the protein with accuracy of 1.0 I, which is 0.1 nanometers. Protein metrics. On the poster benchmark, AF3 achieved a success rate of 76%, far exceeded represent a 2 times improvement in accuracy. That's insane. That's such a huge improvement. Feature 2, protein nucleic acid interaction prediction. DNA and RNA nucleic
nucleic,
nucleic
nucleic,
nucleic
nucleic,
nucleic. DNA and RNA are like the cell's blueprints, and proteins read and edit these blueprints. AF3 can predict how a protein's hand grasp specific locations on DNA or RNA. Concrete examples: bacterial transcriptional regulator, CRP, binding to DNA. Complexity contains protein, DNA double helix, and a small molecular, cGMP. Accuracy scores 82.8. That's really high mark. Feature 3, antibody-antigen interaction prediction. Antibodies are like the immune system's missiles that need to precisely identify specific targets, antigens, on viruses or bacteria. AF3 can predict how the antibody's warhead binds to the target. Example: human coronavirus OC43 spike protein with neutralizing and antibodies scale 4665 amino acid residues, heavily glycosylated. Accuracy, full complex. Performance improvement. Almost doubled. More than doubled. That's insane. Feature 4, covalent modification prediction. Proteins and nucleic acids are often decorated with additions like phosphate groups or sugar chains. These modifications are like gold planes, machine parts, changing their function. AFS can predict structures with these modifications. Modification types. Phosphorylations, glycosylations, methylations, bonded ligands. Performance data. Wow, that's insane. Function 5, large-scale complex prediction. Many cellular function requires multiple proteins to assemble like Lego blocks into giant molecular machines. AF3 can predict ultra-large complex contains thousands of atoms. For technologies, AlphaFold 3 employs a diffusion model architecture, a major innovation compared to AlphaFold 2. The entire system consists of 3 core technical modules. Imagine looking at a giant relationship network map, where each dot represents an amino acid or atom, and the lines represent potential interactions between them. Performer is like a relationship analyst that continuously updates this map, discovering which dots have the strongest relationships and which can be ignored. Through 48 iterations, each round examines triangular relationships. Let's each dot pay attention to other important dots. Updates the entire relationship network. The process is like analyzing friendships on a social network. Initially, knowing only direct friends. After multiple rounds of analysis, discovering friends of friends, common interest groups, and other complex relationships. Technical details. Input. Cell representation. Single representation. Call operations. Triangle updates. You see, here it doesn't give English translation here. It's just give Chinese. That's not what I'm expected. So cooperations is triangle updates, triangle attention, transition. I still feel it's a little bit confusing to understand. Comparison. AlphaFold 3. Use MSRA module. What is MSA? 4 blocks plus Performer. The trunk network is Performer, 48 blocks. Structure prediction, diffusion module. Parameters, 500 million to 600 million. Training time, 150K steps. Okay, support moleculars, everything. That's insane. Technology and concepts. Okay, here is MSA. Multiple sequence alignment. Arranging multiple homologous sequences together to identify conserved and variable regions, revealing evolutionary information. Diffusion model. Denoising diffusion probabilistic model, a generative model that creates data by gradually adding and removing noise. LDDT, local distance difference test, a metric measuring local similarity between predicted and true structures. Token. In AlphaFold 3, a token refers to the basic unit processed by the model, which can be an amino acid residue, a nucleotide, or a ligand item. Terminology 5. SMILES, simplified molecular input. Line entry system, a standard for representing chemical molecular structure using ASCII strings. Interesting. C means carbon. CC means acid. Equal means double bond. C equals C means acid line. Triple bond, pond. C pond C means acetylene. Brackets means branch. CC, branch CC means isobutant. Numbers means ring closure. C111, C1, CCC1. Okay. Symbolism. Legend. A small molecular compound that can bind to macromoleculars like proteins. Includes inhibitors, agonists, cofactors. I have some friend who study in medicine, especially in United States. And one of the complaints they have is that, those English words for the chemical materials are very complex. Now I understand it. Epitope. A specific region on an antigen molecule that can be recognized and bounded by an antibody. PDB, Protein Data Bank. The world's largest database of 3D structures of biological moleculars containing over 200,000 experimental determined structures. glycylation. A post-translational modification process that adds sugar chains to proteins or lips. Okay, here's some metrics. Docking ecology. A comprehensive metric for evaluating protein-protein interface prediction ecology. The equation may pronounce as docq equals open parenthesis f net plus f non-net plus f RMSD close parenthesis divided by 3. System design. AlphaFold 3. So you see, it doesn't provide an English translation here again. 6 layers. Input layer, protein sequence, RNA, DNA sequence, ligand, smiles, covalent bonds, feature processing, input embedder, template search, MSA search, conformer generation, network trunk, template module, MSA module, performer, structure generation, diffusion model, recycling, 3 cycles, 20 diffusion iterations, confidence estimation, output layer, 3D atom coordinates, PLDT scores, PAE matrix, detailed module design, input embedder. So you see, it doesn't provide English translation here as well. Processing pipeline, sequence encoding, positional encoding, feature fusion, output dimension, single representation, and pair representation. Template module. A long 3D structure that is homologous to the target sequence and can serve as a reference for prediction. Target sequence, HH search, PDB database, template module, MSA module. It doesn't provide English translation again. NAF, effective number of sequence. Calculation, number of clusters after clustering sequence at 80% identity. I found that even if I read it, I still don't understand pretty well. What's wrong with my understanding? Because I'm stupid? Why? Performer. A. Innovation. Triangle. Multiplication. MSA3. Triangle multiplication. 48 blocks. Performer. AlphaFold 2. Evil formal. Speed up. Diffusion module. 3-layer architecture. Atom. Input noisy coordinates. Add conditioning. Layer 2 global token. All tokens. Global token. All tokens attach to each other. Sequence local. Atoms within same residue attend. Output denoised coordinates. You say it's. I guess that I need to emphasize it again. Ensure for all Chinese all contents, it includes the report provide both Chinese description and English description. Yeah, I think that's important. Layer 1, pre-atom conditioning. Layer 2, global attention. Layer 3, sequence local. Training pipeline. Training is divided into 3 stages. Initial training. Parameter, data sampling strategy. Fine-tuning. Stage 2 and 3, fine-tuning. Purpose. Pipeline, inference pipeline. Inference. It's the process of using a trained model to make predictions on new data. Inference workflow. Input preparation. Forward pass. Sampling strategy. Ranking and selection. System design. User input. Feature extraction. Input embedding. Embedder. Template module. MSA module. Performer. Diffusion. Confidence. 3D structure, PLDT, PAE, major milestones. It published May 8, 2024, published in Nature. Citation growth. Now it's already January 2025, but it even didn't get the data, but user projected. So you see, there's some hallucination. Open source and the community. Commercial application. Isomorphic labs. Demis Hassabis. How to pronounce?
Demis Hassabis.
Demis Hassabis.
Demis Hassabis.
Demis Hassabis.
Demis Hassabis.
Demis Hassabis. Application. Target identification. Visual. Virtual.
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 Virtual. Virtual screening. Lead optimization. Academic impact. CASP. Critical assessment of protein structure prediction. Follow-up citation. PROTAC. Proteolysis targeting chimera, a class of bifunctional small molecules that can simultaneously band a target protein and E3 ubiquitin ligase, including target protein degradation. Timeline. Eight jointly supervised author and 7 equal contribution. John M. Jumper, Senior Research Scientist, Google DeepMind. D means Hasabi. AlphaGo, AlphaZero, AlphaFold 2, Gemini. That's insane. H index is 107. That's horrible. Ah, academic lineage. Hinton to Hasabi. Baker to Jumter. Zisman to Germsburg. Investor perspective. Competitive landscape. Isomorphic lab.
Isomorphic.
Asymorphic.
Isomorphic.
Asymorphic. Recursion pharma. Acyclical medicine. Schrodinger. Relay therapeutics. Generate biomedicines. Insights and implications. Scientific insights. Number one, power of unified frameworks. General, bigger and specific. Multimodal synergy. Insights 2. You see, doesn't have any translation again. Diffusion model. Discriminative. Generative GAN. generative VAE, diffusion, chemical diversity, multi-scale learning, no handcrafted features. Insight 3, data still the bottleneck. Synthetic data, active learning, transform learning, methodology insights. Less is more. Big data. Insights 2, cross distillation,
Distillation.
distillation,
Distillation.
distillation. General methodology, discriminative, generative, distillation fusion. Industry impact. Traditional power diagram to AI-driven power diagram. Oh, that's crazy. AlphaFold effect. Emerging opportunity at first drug companies. AI first, AI tool platform, data companies. Return industry, CRO, structural biology, traditional docking software. Chemi information companies. Where the relationship between AI and human experts. Tool revolution will lead human role change. Human AI collaboration. We have a major called human computer collaboration, HCI. Maybe we will have a major in future called as HAC, human AI collaboration. Future outlook. AlphaFold 4, technical evolution, dynamic modeling, function prediction, de novo design, multi-scale modeling. Limits of AI in biology. So AI is a powerful tool, but it's not about replacing scientists. Future belongs to human AI collaboration. The biggest breakthrough came from humans' creativity and AI's computation. Okay, let's see. DeepMind. No fragment. Yannick Fletcher. Andrew Capacity. Outreach I biology. Mm. Mm. MIT open course. Wow. Tremendous paper. Stat question. 3 blue, 1 brown. Okay, I already subscribe most of those YouTube channel, some Chinese models and resources. So conclusion, AlphaFold 3 represents a giant leap in AI application in science. It not only inherits AlphaFold 2's success in solving the protein folding problem, but further extends prediction capabilities to almost all biomolecular types. Insights for computer science students. Interdisciplinary matters. Fundamental matters. AI is not omnipotent.
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 Omnipotent. Impact takes time. The story of AlphaFold 3 is far from over. It's just the beginning of AI-enabled scientific discovery. In the next 10 years, we might say, that's amazing. Yeah, well, I guess that this report is easier to read than the original, paper. But still, maybe because I lack of biology background, so it's actually still a little bit hard for me to really understand the whole paper. Even though I guess I will still keep doing this, which means I will try to keep reading academic paper every week, and let's see how it works 12 years later. Whether I feel more comfortable to read any given new academic paper. Yeah, that's pretty much for today. Thank you. Bye.